Hello everyone, in this presentation we will be talking about organic barley production. Under this topic we will be looking at the introduction, crop improvement through breeding, agronomic practices of growing organic barley for food, feed and malt, harvesting, threshing and storage, malting. You will also find some of the sources or reading list for this presentation. Introduction. The crop is commonly known as barley. It is locally known as Na in Dzongkha and Femong in Shashro. It is scientifically known as Hodium species. It belongs to the grass family Poaceae. The wild relative of the crop is believed to be Hodium spontaneum. Barley is one of the world's oldest crop plants. Globally, it is the fourth most important cereal crop after wheat, maize and rice. Barley is highly adaptable compared to other small cereal grains. Therefore, it is mostly grown in the regions or areas where rice and maize are not grown or where they cannot be produced. One of the features supporting its extension into almost extreme regions is its ability to resist dry heat compared to other small grains. World production. In 2004, it was reported that about 70 million hectares of global area are under barley production. Globally, 160 million tons of barley production has been reported in 2004. The developing countries account for about 18% or 26 million tons of total barley production and 25% or 18.5 million hectares of the total harvested area in the world. The average productivity is around 2.4 ton per hectare. Some of the major producers are European Union, Russia, Ukraine, Canada, Australia and USA, forming about 75% of the world production. It is cultivated in almost all parts of the world except in the tropical regions. Barley is an important food for people living in the cooler, semi-arid areas of the world. In general, barley is more productive and its yield is more stable against seasonal variation or changes than wheat and most of other small grains. Types of barley. There are three types of barley, namely Hodium vulgare, a six-rowed type of barley, or commonly known as six-row barley, has a spike notched on the opposite sides with three spikelets on each notch. At each notch, there is a flower or floret that later develops into a kernel. Hodium distinctum. A two-rowed type of barley, or commonly known as two-row barley, has central florets producing kernels and it has lateral florets that are sterile. And three, we have Hodium irregular, commonly known as irregular barley. It is the least cultivated with fertile central florets and varying proportions of fertile and sterile lateral florets. Origin and distribution. The origin of barley remains unclear. However, archaeological evidence of barley cultivation has been found dating back to 8000 BC in Iran. It has also been indicated to have been farmed in China around 7000 BC, in Spain by 5000 BC, and in England by 3000 BC. The ancestor of the barley is believed to be the two-rowed barley as we have already seen earlier. Uses. It is commonly grown as an animal feed, which comes about 70% in the world production, both fresh and as grains. About 16% of the world production is also used for manufacture malt, seed, and in other industries. 14% of the world production is used for human consumption in various forms. Uses of barley in Bhutan. The flour is used for consumption as well as for ritual ceremonies. The grains are also used for brewing alcohol and is also commonly grown as winter feed for cattle. Crop improvement through breeding. Intensive breeding has been done to improve performances and quality of barley grains, which has resulted in the reduction of genetic diversity in the elite or improved cultivar. Ionizing radiation and chemical mutagens have been used to increase mutation rates since 1927. According to World Health Organization (WHO) in 2016, ionizing radiation is defined as a type of energy released by atoms that travels in the form of electromagnetic waves such as gamma or X-rays or particles such as beta or alpha. Chemical mutagen is defined as a chemical substance that can change the genetic material of an organism or an agent that alters the DNA sequence. Until the mid-1900s, conventional crossing was used to develop new cultivars. Since 1950s, more advanced plant gene technologies have been used. These advanced technologies can reduce the time between improvement and release of a new variety, meaning the accessibility of the farmers or producers of such improved varieties are increased. Most of these improvements are targeted at disease resistance and malting quality. For example, marker-assisted selection and whole genome breeding are used. Ashraf, Akram, Rahman, and Fulat in 2012 have defined marker-assisted selection or MAS as the process of using morphological, biochemical, or DNA markers as indirect selection criteria for selecting agriculturally important traits in crop breeding, in their journal titled Marker-Assisted Selection in Plant Breeding for Salinity Tolerance. One such example of a variety developed through these technologies is the flagship variety that was released in 2004 in Australia. Molecular techniques or embryo rescue have allowed using wild relatives as a source of genetic variation. Similarly, Xian in 2012 in a paper titled Molecular Markers and Marker-Assisted Breeding in Plants has defined molecular breeding in a broad or general sense as the use of genetic manipulation performed at the DNA molecular levels to improve characters of interest in plants and animals, including genetic engineering or gene manipulation, molecular marker-assisted selection, genomic selection, and more. There are three gene pools in the genus Hodium: primary, secondary, and tertiary. Primary gene pool, that is Hodium vulgare subspecies spontaneum, has no cross-ability barriers with the cultivated variety, subspecies vulgare, due to their astounding morphological similarities. Since the crossing is between the same species, hybrids are fully fertile with normal chromosome pairing, which means they are capable of producing seeds. When these two species are grown together, spontaneous or natural hybridization occurs sporadically or occasionally. This gene pool has been primarily used to develop or improve disease resistance and abiotic stress tolerance in the barley crop. Secondary gene pool. Hodium bulbosum is highly self-incompatible. The genomes of Hodium bulbosum and Hodium vulgare are genetically closely related. Thus, they can be readily crossed under artificial or controlled conditions. However, seed setting can be very low and their multiplication is limited. Tertiary gene pool. It has 30 species, making it quite rich in diversity for barley crop improvement. However, they have strong cross-ability barriers, making it difficult to use. For example, the hybrids are almost totally sterile, and the genome of the cultivated barley does not readily recombine with other species, because of which the genetic material usually does not exchange.